Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody back to In the Interim. Today I have a really cool topic. Topic is response adaptive randomization. And I'll talk a little bit about the structure, but I have a guest with me today, an expert in clinical trials, an expert statistician. Michael Proschan has been at the NIH for 19 years. He's a mathematical statistician in the Office of Biostatistics Research of NIH, and specifically NIAID, the Institute for Allergy and Infectious Diseases. And Mike has 3 books to his credit: Essentials of Probability Theory for Statisticians, Statistical Monitoring of Clinical Trials, and Statistical Thinking in Clinical Trials. So welcome to In the Interim, Mike.
Thank you very much. Good to be here.
All right, so Mike and I are probably known to be on opposite sides of response adaptive randomization. Mike and Scott Evans wrote an article, Resist the Temptation of Response Adaptive Randomization. I've used response adaptive randomization. So I thought it would be great if we talked about what do we agree on, what do we disagree on, what are the issues around response adaptive randomizations. Are they good? Are they bad? Are they ugly? And hear what we agree and disagree on. You sound good, Mike?
Yeah, no, that sounds like a great discussion.
Okay, so why don't you, for our listeners, tell us what response adaptive randomization is?
Yeah, response adaptive randomization is changing the randomization probabilities in response to outcome results. So you're seeing the actual outcomes, not just changing randomization probabilities in response to covariates, baseline covariates, but actually looking at outcomes. And so the idea is that you can assign more people to the better performing arm, and in that way, perhaps be more ethical.
Okay, so that the non-response adaptive randomization in a trial might be that there are 2 arms in the trial, and you do 1-to-1 randomization from the start of the trial to the end of the trial, regardless of what the data show. Response adaptive randomization, you have planned interim analyses, and which of the 2 arms is doing better, you might assign differentially with a higher probability. It might go to 60%, 70%, 80%. In a multi-arm trial with either multiple doses, you could change the randomization to favor those arms during the course of the trial. So that's the response adaptive randomization. Okay, so on the side of response adaptive randomization, and so maybe you'll resist the temptation. So what is it about response adaptive randomization that you find to be a negative?
Yeah, so one of the big problems that I have with it is that what can happen is that as you're changing these randomization probabilities, let's suppose you're in an extreme situation and you're now assigning 90% of participants to the active arm. Let's say there's just two arms. Then what happens is if that happens later in the trial, there could be temporal trends that occur in clinical trials, and they do happen. And maybe the earlier patients are different from the later patients. And so all the later patients, or most of the later patients, are getting assigned to the active treatment, but the later patients are also tend to be healthier than the earlier patients, for example. And so if you're not careful, you could, declare a treatment benefit when it's actually just a temporal trend. And there are ways to try and deal with that, but that's one big concern. I would say that's my main concern. I also, though, think, like in an unblinded trial, if patients are seeing who's in the, if patients are seeing these randomization probabilities changing, then they get good knowledge of, which arm is doing better. That could cause patients who are in the other arm to perhaps drop out or start taking the active drug and changing their behavior, basically. So that can influence the results of the clinical trial. So I would say, both of those are concerns. My main one is the first one, because most of our trials are blinded. Although I must say that in these multi-arm trials, there are several of them happening during COVID, in which you figured out which arms you were eligible for, and then you got randomized to one of those eligible arms, and which of those treatments you were eligible for wasn't blinded, but whether you got the active or the placebo was blinded. So sometimes there is some level of unblinding in these trials.
Yep. Okay. So let's let's unpack a little bit the temporal trend. So the issue could be that in the first half of the trial, it's largely sicker patients. You're doing 1-to-1 randomization. The treatment is doing better. You have two arms. You have a placebo and a treatment. The treatment's doing better. You change the randomization 75%, 25%. And in the second half of the trial, the patients are relatively more healthy. When you get to the end, if you just take the raw average of the treatment, you have more patients during the healthier part, and the raw average is going to look better than it is on the placebo in that scenario. Even if there's not a difference between them, the temporal trends can be different. Now, you alluded to, you can correct for that. So if I were involved in a trial where we had that, you could put in a covariate for the time. And that covariate's going to pick up, because you have the same arms in the first half and you have in the second half, that the patients are less healthy because they're more healthy. They do better in the second half because you have both the arms, the data, and you make an adjustment for your relative effect of that. It would be a way to potentially mitigate the temporal trends in that scenario.
Yes.
Now, that means at the end of the trial, you need an adjusted estimate. You can't just take the raw averages of that. So now you've got an adjusted estimate. In many clinical trials, we adjust for covariates. We adjust for gender. We adjust for age. We adjust for severity, other aspects of this. So that could be a way to mitigate it. The unblinding, I agree, is an issue. If an investigator sees that all of a sudden a particular treatment is coming up more and more, and they know it's response adaptive randomization, that could potentially lead to maybe not even enrolling a patient in the trial, because boy, that must be doing better. I'll just give that outside of it and other aspects of it. You can have trials that are completely blinded, and the randomization can shift and nobody's the wiser, other than the people that are analyzing the unblinded aspect of it. Now, you didn't bring up the part that I thought for sure you would bring up, which is the first criticism, is the lowering of power. Even if we do that adjustment, whether you're comfortable the adjustment in a two-arm trial, if I'm trying to compare the two arms and say which one's better. Now, there are weird scenarios in terms of the variation of the arms where you want differential randomization, a 5% rate to a 25% rate. You'd rather have more on the 25%, but let's ignore those. Almost always you want equal randomization of the two arms.
Yes.
And so going in your case of 9-to-1 or 3-to-1 or 4-to-1 lowers power.
Right. Yeah. I was going to Yeah, I didn't say that because I did say that, as you mentioned, that there are ways of dealing with this. I didn't mention their drawbacks, but like one of the standard ways of doing it would be to stratify by time, and then within that second half, if you've got 9-to-1 randomization, you're very inefficiently estimating the treatment effect because of that unequal randomization. Yeah, so
Right.
I was going to say that in response to how you try and fix these issues.
Okay,
Yeah.
so you're setting me up, is what you're doing. What you're just saying. Yes.
I would
Yeah.
never do that, Scott.
Okay, so I think we largely agree on these points. I agree it lowers power, and I think if you're running an experiment comparing two arms, that you need another reason besides statistical for doing response adaptive randomization. We'll come back to the ethical issues and what you think about that in this scenario. And largely, it's two-arm trials. There needs to be a compelling reason to do it under those circumstances.
Yes.
On that, the unblinding, I think, is an absolute issue. If you're in a scenario where there's unblinding, and there are a number of scenarios where patients and investigators are unblinded. Oncology trials, they're frequently unblinded. It's very hard to blind the chemotherapy regimen or the intensive regimen that they're getting in oncology. They can be unblinded. And there are some trials that it's very hard to blind patients and investigators in those scenarios. But there are certainly many that can be blinded. So the temporal trends, temporal trends, I think, in your life of being an infectious disease is certainly a bigger issue than in other non-infectious diseases, in ALS or Alzheimer's or oncology or other scenarios where you might not think time makes as much of a difference. Now, it can make a difference in those scenarios. In severity, in the trial, the behavior, the sites you open within the scenario. But certainly, I imagine in infectious disease, temporal trends are a much bigger issue.
Yes, and that's true, especially with a new disease where temporal trends could result from you're just doing better background care. You didn't know how to treat them, before, and now over time you're learning, or there could be a new vaccine introduced that changes things. And there are a number of other things, viral variants, change. And so really, I mean, that's a problem, even if you don't want to talk about response adaptive randomization, that's a real problem when you do a trial and then, a couple years later, everything has changed and you're
Right.
wondering whether, whether you can really rely on the results. So,
Right.
yeah, we're in a particularly tough situation in infectious diseases.
Yeah. So we brought up the idea that suppose we're doing response adaptive randomization and we're going to adjust for strata.
Right.
That adjustment is valid and very reasonable as long as the effects are additive. So we're fitting a logistic regression or a continuous endpoint on that. As long as that healthier, you brought up the example that they're healthier in the second half. If we make an adjustment and the relative treatment effect stays the same, it's just patients go up and down equally in the two arms, that's a perfectly valid thing to do. Interactions with time, which you just brought up, the potential that the disease is almost different in the second half, it's a new variant, is clearly a problem. But it's clearly a problem to science that you might run a trial and then wonder 6 months from now, is our treatment still valid? In the scenario of that, in that. So that's a huge problem. Okay, now let's talk about, so two-arm trials, I think we largely agree on that. Let's talk about multi-arm trials. So what's different about multiple arm trials? So, and it's not uniformly that response adaptive randomization is better in multiple arm trials. It really depends on the goal of that. So we've done platform trials where there are 5 companies that bring their drug in, and they would all like to understand in a phase 2 trial, does their drug work?
Yep.
That's one potential platform trial, and we'll come back as response adaptive randomization good there. Another potential trial is where you have multiple arms and you're really interested in the best arm. And the goal is to find the best, almost a king of the hill platform trial of that. Now, one of those is perhaps dose finding. So we've done a response adaptive randomization trial where we had 7 doses in a diabetes trial, where we're looking at endpoints like HbA1c, blood pressure, heart rate, weight loss over 7 doses. And in that case, the company, the sponsor, they want to know the best arm. So if we do response adaptive randomization in a multi-arm setting where our goal is to find the best and probably identify how good it is, you can imagine by lowering the probability to arms that are early on identifying themselves as not as good, I increase the randomization to the better arms. And in simulations, in certain scenarios, in certain ways to do response adaptive randomization, that can improve your ability to find the better dose and to identify the better dose. Now, that's at the negative to doses you go away from. You learn less about the doses you go away from. So coming back to a platform trial where 5 different sponsors put their drug in, if I'm doing response adaptive randomization and I'm favoring company A, it's to the detriment of company B, perhaps.
Right.
And that might not be why I'm doing the trial in the scenario. But in a scenario that matches these goals of king of the hill, find the best, identify the best, it can improve power. Is that reasonable?
Well, so I will stipulate to the fact that that there are some situations where I find the idea attractive of trying to assign more to the arms that are doing better. And like you said, that may help you find, a good drug fast, which, might be especially important in certain situations where there's nothing that works and, it's very serious disease. So I can see being attracted to it. But as you say, it's to the detriment of these other drugs. And the problem is, if the early results are not necessarily, that reliable. And, the late Richard Simon once said, hey, we don't even get good estimates of the treatment effect at the end of the trial. And now you're trying to respond to, estimates that you're getting very early on. So I think one of the things that I imagine that you and I both agree on, I think we've already hit on one, but I think another one that we both agree on is that if you're going to do response adaptive randomization, you should have a burn-in period of a more traditional randomization first, because those early results could be quite variable.
Yeah, absolutely. And for example, Thal and Waffen presented response adaptive randomization goes bad when you lessen the probability on the control. So in my 7 doses in a diabetes trial, if I'm lessening control in that scenario because it's doing worse, my ability to identify those doses goes the wrong way. It's kind of the 2-arm trial scenario like that. So most of the time we do response adaptive randomization, we're trying to fix the control and we're altering the other ones in that scenario. And yes, adequate burn-in. We're doing lots of simulations to find what's the likelihood I'm misled and I put too many on that. And that's part of this is I'll agree absolutely that you can do it poorly. Right. I've been involved in a couple trials that have gone a bit awry, but you can have algorithms that don't do it well. And you can have algorithms that do it well, that increase your probability of finding the right dose and all that. So we've done trials on Alzheimer's where we've had 5 different doses, diabetes with 7, even sepsis, which I think is an infectious disease. Is that an infectious disease? I know it's an infection of something causes sepsis typically, but that multiple treatments of that. Now we did it in COVID, and I'll come to that. I think that I'm teeing it up for you in COVID in that scenario. But first, I want to come back. I did not hear Richard Simon passed away.
Yes, he did.
Oh, that's awful. He's
Yeah,
certainly a hero to statisticians and a
absolutely.
brilliant guy and a wonderful guy. Yeah.
Yeah.
Huh. That's too bad. So in a scenario where multiple arms, if the goals of the trial, if you're okay, lessening the lower dose, lessening the higher dose because it's got adverse events in this, and you're good with the less precision on that. If done well, that could potentially lead to improvements in multi-arm trials. We're at least in the same neighborhood.
Yeah.
Okay.
I mean, I do see the potential for some benefit of that. Yes.
Yeah. So now let's talk about a multi-arm trial that you were involved in, and this was absolutely incredible trial. The PALM trial was in a platform trial in Ebola, one of the first platform trials in infectious disease, if not the first, perhaps. Incredible effort by the NIH. And you investigated 4 different therapies for the treatment of Ebola.
Right.
And this was not the sort of 2014-15 go around where this, but I think this was later, sort of 2018-19-ish, where you investigated, right. So investigating, and I should probably, I wrote down to make sure that you investigated within this ZMapp, remdesivir, MAB 114, and a Regeneron EB3.
Yes, that's right.
So within this, so 4 of them. I think in the way you set it up, ZMapp played a role of a control.
It did, yeah. And there was some criticism about that because ZMapp, in an earlier trial in Western Africa, the trial was stopped before the evidence was sufficient to say that it's, better than not giving anything. But we felt pretty confident that if something beat ZMapp, then it would have to be better than giving nothing.
Yep. Okay. And so the results of this trial, and largely you compared each of the other 3 to ZMapp. Each of the other 3 drugs were to ZMapp. And at least in the part that I have within this, the remdesivir was stopped because it didn't improve upon ZMapp, but the other 2 arms, approximately 50% mortality within 28 days on ZMapp and remdesivir, and approximately 35% mortality at 28 days on the other 2 drugs.
Right. Yeah, so there was a clear separation between ZMapp remdesivir pair and the other 2, the monoclonal antibody 114 and the Regeneron product. It was quite clear that those 2 were better than ZMapp and better than remdesivir.
Okay, so if we were to take those sort of results and say, what if we would have done response adaptive randomization? And I'll come back and ask, was this ever entertained, or all of that? Reasons, yes or no. That if you simulated a scenario where the control was 50 and you had 2 arms that were 35 and another one that was 50, and you did interim analyses, and you simulated it where it changed the response adaptive randomization to those, you might find that you're quicker to find one of those 2 were effective, and you maybe put more people on there if you simulated that scenario. Now, I don't know the goals of this trial and all of this. I wasn't there, but was this entertained? Are there reasons not to do it there, reasons to do it in the trial like that?
Yeah, so that's an interesting trial because that was not a trial in which, you're eligible for these, and so we're going to randomize you to one of these two instead of one of these four. That was a trial where you had to be eligible for all four. And
Yep.
so, that's a situation where I think I would, I would object less to response adaptive randomization. I still would not do it because I think that there are, things that can go very wrong, and I think you can achieve similar results by, by monitoring and perhaps responding, stopping arms early. And they were, it was stopped early, not super early, but it did not go all the way to the end. And so, so I think you can achieve similar things by having equal randomization, and then you would get better information on, on both the two products that did well, the monoclonal antibody 114 and the Regeneron product. I don't know that, with response adaptive randomization, perhaps it would have given more people to only one of those arms. I didn't do any simulation after the fact to see what would have happened if we had used response adaptive randomization. I'm guessing that you may have done that, Scott.
It's not like I'm going to spring them on you, but it is actually the case. I think we did a number of these during Ebola with a Gates trial that never enrolled any patients in this scenario. The sort of the somewhat optimal circumstance for response adaptive randomization would be if one of those four was that effective, 35, and the rest were 50. With two of them, you do find advantages in that scenario. And it's sort of a question of how important is time. And you touched on that within a pandemic. Within a pandemic where maybe time means a huge thing, getting results out earlier on one of them, how important that is relative to getting results on all of them, for example. And I think you're right. So about 100, I know you're right, about 175 patients each to these arms, roughly, in that. And then you stop two of them, which is kind of a brute force response adaptive randomization where it goes 1 to 1, and then it goes 1 to 1 to 0 to 0, because I think you kept enrolling the other two.
Yeah, so we made the decision, the DSMB made the recommendation to continue follow-up, because not everyone had had the full 28 days of follow-up, to continue follow-up, and then the final results would be after the people that had already been randomized, were evaluated for that 28-day outcome. That was the final analysis.
Yep. Okay. So we did the REMAP CAP trial does response adaptive randomization. It's a little bit, is it a multi-arm trial or not? It has domains, so it actually does multifactorial randomization to a patient. During the height of COVID, I think one patient was randomized to 7 domains. What are the domains? Where they're therapeutic anticoagulation arm, an immune modulation arm, there was a vitamin C arm. Many of these are thought of as fairly, you need to make a decision in routine care. Do we give them? Do you give a steroid, yes or no, aspect of it? And so in that setting, some of those domains are multi-arm domains. The immune modulation had anakinra, tocilizumab. it had interferon, and it had no immodulation. And so that was one where actually tocilizumab and saralimumab, IL-6 receptor antagonists, got higher randomization faster. Then we had domains that were 2, and particular one of them, simvastatin, ended up randomizing at about 8-to-1 or 9-to-1 at some course of it when COVID stopped in that setting. In hindsight, we have changed the design that we only allow, I believe it's now 70%, so 7% to 30%. It's a little bit different. That it's a little bit of multi-arm and 2-arm, where there's a question of, do you give a simvastatin, yes or no, was the randomization. So you could call that a 2-arm domain, but at the same time, interactions between the different domains became really quite important. Does a statin interact with IL-6? Does it interact with steroids? Does it interact with antivirals? Things like that. And so putting more patients on the things that were doing better was perceived to have benefits in that scale. We didn't want that to happen, so we've put governors on it in this one. But we have done in infectious disease that within COVID in that. Now we're doing it in community-acquired pneumonia, which is certainly an infectious disease and can have seasonal variability as well.
Right.
Okay. Another trial, and then I want to come back to the question of ethics. You brought up ethics and your views on that. So I-SPY2, are you familiar with I-SPY2?
I know something about it. Yeah.
Yeah. Okay. So this is a neoadjuvant breast cancer trials, phase 2. It was a platform trial. And what's a little bit different than some other platform trials, multiple agents come in, there's a standard of care. And in breast cancer, they really differentiate breast cancer really well. Baseline factors on estrogen receptor status and hormone receptor status. And so there's mammaprint status, but it plays a lesser role. But largely, you can think of the 8 classifications of plus minus for those. That in the trial, even though it was multiple sponsors who brought a drug in, Amgen, Merck, brought drugs in, that we did response adaptive randomization because it was believed efficacy was likely to vary by the 8 subgroups. So if one drug was being favored within hormone receptor positive, the other drug might be favored in HER2 positive, and that drug's getting more patients within subgroups that they're doing better on, and less where they're doing worse on if we keep up equal randomization. So in a circumstance where there's a platform trial and sponsors were good with this plan, even though it kind of goes back to this platform trial idea that it's multiple sponsors, it's almost this personalized setting in a case where there's strong belief that these diseases are largely related but different. Okay, wildly shaking your head that's awesome and you love response adaptive randomization. No. Yes.
No, I didn't mean to convey that, Scott.
Okay. So let me, I don't want to this, so I brought several trials up within this. Let me make sure you want additional topics to bring up before we touch on ethics of this.
Well, I do think one topic, and you mentioned a little bit about this, is that the more complicated the randomization, the easier it is to make an error. And I certainly worry about and the same thing happened with covariate adaptive randomization. There was a trial in which, they meant well, but the randomization scheme that they used didn't maintain the same, different patients, depending on when you arrived in the trial, you had different probabilities of being assigned to, a given arm. And that led to problems. And so I worry that, if you have people who are, want to go out and try this new response adaptive randomization, they might not be aware of how careful you have to be because you can mess it up. So just the mechanics of it, I think, are more difficult.
Critically important that operationally it's done well. You need data availability. We didn't touch on the notion that you need your endpoint proximal enough that you can respond to it. In the Palm trial, for example, 28-day mortality, I think largely patients by day 10 have probably determined not.
That's
could
right.
be a
About
possibility.
About 95%, if they died within 10 days. So
the other side that this has been presented is the ethical side of this. That if you're a patient in a trial, and it's been phrased, and my father, Don, whose original introduction was multi-arm bandit problems, where you phrase this as there's a horizon of patients and you're trying to have better outcomes for all of them, and the patients in the trial and the patients outside the trial. So part of this depends how big is the horizon outside as opposed to inside. If every patient in the world has the disease and is in the trial, you might behave differently than if you have a very, very small fraction and what you learn is going to be given to millions of people afterwards within this. But the idea is that during a trial, if you're doing 50-50 randomization, if there was a VIP who could see the data and now had to be treated and make a decision, that person would be treated differently than a patient in your trial. And is there an ethical advantage? Is there advantage to patients to have the increased probability, say in a multi-arm trial, the two-arm trial? Now, two-arm trial is a little bit weird because you're lowering power and what that means ethically. So it's a complicated story ethically.
Right. Yeah. Yeah. No, as many trials are, it's a difficult issue because you definitely, I feel, it makes sense that if I'm a patient, I want to get the best treatment. But, I'm not sure I am getting the best treatment if it's made on the basis of interim results. We've talked about the fact that, you want to make sure you have conventional randomization, up to a point so that you don't have that. But I mean, that's, one of the things, every time I bring up the E word, the ECMO trial, people go crazy because, ECMO was a trial involving primary pulmonary hypertension, and it was an early example of response adaptive randomization. I grant that a lot has been learned from ECMO. But that did lead to a severe imbalance where 11 people were given the new treatment, and one person was given the standard treatment. That one given the standard treatment died, and the conclusion was made that ECMO was better, which turned out to be the correct decision after another trial showed that. But that caused a lot of problems, and some of the problems it caused we haven't touched on at all because I don't think that these are major concerns for large trials. But we take for granted that the sample size is a fixed quantity. We condition on the sample size. We never have to take into account that is a random variable that needs to be taken into account. But in that ECMO trial, you do have to take that into account, the fact that ECMO, that the sample sizes themselves are giving you information about whether the treatment works or not, and in particular, the lopsided results, indicate that the treatment must have been working because, otherwise it wouldn't have gotten lopsided. And so, it's not even clear how to analyze the data. Now, if you're a Bayesian, then you say, no problem. But if you're a non-Bayesian, there were many debates about how you could even analyze those data. Very interesting debates. A nice paper by Colin Begg, after that trial. So it was a fascinating trial. But I think those are some of the issues that we haven't really touched on that also give me some concern. As I say, I think that those are not particularly problematic with large trials. I'm not sure how large they have to be to not have them be a problem. But, who would... Yeah, go ahead, sorry.
Right. So I'm so sorry. This is the Bartlett play the winner design.
Exactly, yes.
Using ECMO as a treatment, and which I think at one point 35 different p-values had been created from that same trial, and it was not clear how to analyze it as a frequentist. Now we won't talk about whether this is about the challenges of sample space inference as opposed to Bayesian inference. But I've never done a play the winner design. I think it is awkward, and I once got the benefit of asking Bartlett. I said, suppose somebody would have simulated this trial and showed you that result. Would you have said, I don't like that, and done something different? He said, yes. If somebody would have simulated that as an example and said that could possibly happen, I would have done a different design, which, I use that as an example of the value of simulation and making sure within an adaptive design that you've seen lots of examples and what this does. And a burn-in here probably would have made a world of difference, I suspect, in that trial. Yeah.
Yes. Or even starting, instead of starting with one ECMO ball in the urn and one standard ball in the urn, if you started with 10 of each or something like that, where it's not so volatile, doesn't quickly devolve into, virtually everyone getting only one treatment. And by the way, that one plus standard treatment patient was objectively the sickest one, as luck would have it. So that patient may have died no matter what the treatment. That baby may have died regardless of treatment. Yeah,
there's a whole separate issue about if you analyze that trial using real-world evidence, other things about whether that rate was there. So that trial is probably the most analyzed trial in the world, I suspect. Very interesting, yes. Okay, so within that, so I think we largely disagree in two-arm trials. Response adaptive randomization is challenging, and you need other reasons why than statistical. Generally not a good idea. Play the winner designs, we largely, it sounds like a neat idea, but it needs to be modified at the minimum to do it. I think there are better ways to do it within the scenario. In multi-arm trials, there are scenarios where as long as it meets the goals of the trial, it can improve some operating characteristics like that. You made the point, a great point, that operations need to be in order to do this well, certainly within it. Unblinding, which by the way was also an issue in that Bartlett trial, that there was unblinding as to the ECMO, certainly needs to be worried about in the scenario if there's potentials for people seeing one of the arms doing better. We both are not experts in the ethical side of it. We think there may be some potential advantages to response adaptive randomization. If I was a patient, I would want to go in a trial that's a response adaptive randomization trial. But I'm not sure we're gonna solve the ethical issue to that. Fair enough?
That sounds fair to me.
All right. Well, Mike, I don't know if you've ever been in the interim before, but I appreciate you being in the interim.
Well, I have not, and thank you for inviting me. This was good discussion.
Wonderful, and clearly to point out the people at the NIH are doing fantastic work. I know it's challenging times, and we all certainly think very highly of the work, and you've had 19 great years at the NIH, and Thanks for your efforts. Keep them up. And
All right. Thank you, Scott.
thanks for joining us in the interim.
All right. Thank you.